Miss Tsukita, don't think I will be able to grade you in Japanese. So only what you have written in English, okay? Same goes for Suzuki Shimbun. Only what you have written in English. Okay, thank you very much. I hope you're reviewing classes at least a little bit after we finish. That will make things a little bit easier to remember. Very quickly, what is the endocrine system? Well, collection of glands that produce and release hormones. They regulate processes of growth and development, metabolism, reproduction. This is a very general idea, right? You can also mention that it interacts closely with the nervous system, influences metabolic activity through hormones. This is very important. And responses usually occur, I mentioned this more than once, occur more slowly, but they tend to last longer than the reactions from the nervous system. I asked you to name only 5 major endocrine organs, just to name them. You have this list of 9, for example. You could have mentioned pituitary gland, hypothalamus, thyroid gland, or parathyroid glands, adrenal glands, pancreatic, islets, the pineal gland, thymus, gonads, any of them, okay? As long as you put 5 of them, it's enough, okay? Of course, this is only the quiz to give you a little glimpse of how you are doing, if you need to study a little bit more on some areas or not. At this point, just to mention them is good enough for me, but that doesn't mean that you don't have to know what each of them does, okay, for the final exam. The blood is composed of plasma and form elements. Here, you can write a lot about them, describing them. You don't need to put everything. Just the main ideas of each of them is good enough. To refresh your memory very quickly, plasma is made up mostly of 55% of the blood, mostly made of water. It's the liquid part. Includes different substances that are dissolved, nutrients, electrolytes, gases, hormones, proteins, and waste products from the metabolism. Helps distribute the body heat. You can mention any of these characteristics, okay? The most abundant solids in plasma is the plasma protein. You may have mentioned, if you want, something about the proteins. These are part of the form elements, the 3 types of form elements that you can find here, the red blood cells, the white blood cells, the platelets. But their names are erythrocytes, leukocytes, or platelets, okay? Try to mention the main characteristics of each of them. Okay, what would you say would be the main idea in the red blood cells? What kind of things did you write for red blood cells? They carry oxygen. Okay, so these are elements that carry oxygen. They're small, you can say these, and they're aneuploidy, so they don't have a nucleus. This is important. And they are very rich in hemoglobin. This is why they can carry the oxygen, okay? Leukocytes, they are complete cells in the blood. They're able to move into and out of the blood vessels. This is important, and we will see later on as part of the immune system. You can also mention the different types of leukocytes, granulocytes, and so on. Okay, this is just to refresh your memory. If you want to go back to these slides from the previous classes. Okay, this was pretty easy, I think, right from the table. Did you have problems filling in when I was asking antigens and what antibodies they are producing? Okay, if I'm type A, then my red blood cells will have antigen A in the surface, so my body produces antibodies against B. Correct? And so on. Then, I'm sure you remember very well from your presentation. Let's hope everybody else was awake during presentation. I took this from your slides, actually your picture as well. The main things that the heart lies between the lungs, it's in the mediastinum. The main 4 chambers, 2 atria, 2 ventricles, okay? 5 great vessels, superior, inferior vena cava, pulmonary trunk for pulmonary veins, and the aorta, okay? This is a big idea, and 4 valves. Remember, between the atrium and the ventriculum going into the great vessels. Okay? So I hope everybody did well here. So Mr. Toriba, you can have the presentation. Let's see. Okay. There you go. You can take your time, please. As always, try to speak slowly. Yes. Don't rush, because you may be a little nervous. So take your time.
Okay.
Yes. Thank you very much. You can start.
Okay. Today I will talk about lymphatic system. Yay! Blah blah blah. Thank you. What is the main function of the lymphatic system? Do you know what the lymphatic system is? Okay, before we answer this question, we will look how the lymphatic system works and the structure, what the structure is. Lymphatic system is an important partner of the cardiovascular system, which we learned last week. We learned last week the capillary exchange. What is capillary exchange? Capillaries are very small, thin vessels. Capillary exchange means the exchange of materials between blood plasma in capillaries and interstitial fluid. Capillary exchange produces lymph. Lymph is the excess fluid or remainder which is left behind by capillary exchange. Okay? Lymph is fluid. Lymph is also called lymphatic fluid. Why is lymphatic fluid generated? Because of the hydrostatic pressure generated by the pumping action of the heart. Almost all of the interstitial fluid goes back into the blood directly by the same route it came out. But small amount of interstitial fluid remains in tissue spaces. The fluid, lymph or lymphatic fluid, enters lymphatic vessels. Lymphatic vessels are also called lymphatics. If there are no lymphatic vessels, we would be suffered from edema. Edema is fluid accumulation in tissue spaces, called in Japanese kushu. And because of edema, the tissue would not work properly. Why? Because the tissues cannot exchange material with blood. Okay, let us summarize the components of the lymphatic system. First, lymph or lymphatic fluid is the excess fluid which is left behind by capillary exchange. Second, lymphatic vessels or lymphatics are the network of tubes which drain lymph. Third, this will be mentioned later, lymph nodes are are the filters which lymphatic vessels go through. Fourth, this will be also mentioned later, lymphoid organs consists of thymus, spleen, tonsils, Peyer's patches, and appendix. You may not know these names, but it's okay, I will mention later. So lymphatic system is the system which consists lymph, lymphatic vessels, lymph nodes, and lymphoid organs. Okay, let us talk about lymphatic vessels. Unlike blood vessels, lymphatic vessels do not follow circular route. Lymphatic vessels are not ringed. It is one-way route. Lymphatic vessels start in the form of lymphatic capillaries. Lymphatic capillaries are thin lymphatic vessels. Lymphatic capillaries have closed-end tips, like this. This is the difference between lymphatic capillaries and blood capillaries. Lymphatic capillaries are very permeable. In other words, even very large, huge molecules like viruses can go through the lymphatic capillaries membrane. Unlike viruses cannot go into the blood capillaries membrane. So, in the past, researchers thought that lymphatic capillaries have open ends like a straw. However, lymphatic capillaries have closed-end tips. Why do large molecules like viruses can go through the lymphatic vessels, capillaries? The answer is they have flaps or flap-like mini-valves. Flap can fold into the tube, so lymphatic capillaries have enough spaces for large molecules to go into the tube. And the flap cannot fold out the tube. Note, so lymph in the lymphatic capillaries does not leak, does not go out. Lymph flowing through the lymphatic capillaries next moves into larger and larger lymphatic vessels, and lymph flowing in the larger lymphatic vessels finally flows into one of the two terminal vessels called right lymphatic duct and thoracic duct. Thoracic means chest area. The right lymphatic duct and thoracic duct, this green one, returns the lymph into the blood in large veins in the neck region. Oh, sorry. And lymph from about 3/4 of the body, this yellow area, drains into the thoracic duct, which is the largest lymphatic vessel in the body. Lymphatic capillaries in the wall of small intestine are given the special name of lacteals, this green one. They transport fatty acid. No. sorry. And maybe these light structures are Pierce patches, which will be discussed later. Okay, it's end of lymphatic vessels. Next, I will talk about lymph nodes. By passing through the lymph node, lymph is filtered. Bacteria, cancer cells, virus-infected cells, and damaged tissue cells are removed and prevented from entering the blood. Lymph nodes do this work by a two-step process. First, debris is trapped by the web of reticular fibers. Second, immune cells, which is discussed later by Ms. Sugimoto, destroy the debris by phagocytosis. Phagocytosis means eating. Note that efferent lymphatic vessels are less than afferent lymphatic vessels. Why? Because flow speed should be decreased for this filtration. If lymph flows fast, debris will go through and flow into the blood. Okay, it's end of lymph nodes. Next, I will talk about lymphoid organs. Thymus is the most important lymphoid organ, I guess. This is located behind the sternum. What is sternum? Sternum is the bone you push to rescue the heart-attacked person before using AED. And thymus is in front of the heart. Okay, what is the functions? First, it is a source of lymphocytes before birth when you are fetus. Lymphocytes are discussed later by teacher. Second, it is a place for T lymphocytes or T cells maturation. If the thymus does not work well, you will be suffered from autoimmune diseases. Even though the thymus is so important, the thymus is a very small organ whose weight is only about 40 grams at the largest period. And the half of the thymus is gradually replaced by fat and connective tissue by age 60 and completely replaced by age 80. This is called involution. So elderly people cannot produce new mature T cells. The second lymphoid organ is spleen. This is the only lymphoid organ which the blood flows through. The spleen is located in the upper left area of abdomen and lateral to the stomach. Lateral means side. The spleen removes bacteria and other debris in blood by physical and biological filtration. The spleen also removes worn out red blood cells and recycles iron in hemoglobin for future use. The spleen is a very large blood reservoir. It can contain more than 500 milliliters of blood. Blood is released in case of hemorrhage. Even though the spleen has many walls and the spleen is the largest lymphoid organ, but we can survive without spleen. For example, if the spleen is damaged by traffic accident and bleeding, a surgical removal called splenectomy may be done in order to stop blood loss, and we can live. Okay. The third lymphoid organ I show you is tonsils. They help protect against bacteria which may invade tissues in the nasal and oral cavities. Tonsils consists of pharyngeal tonsil, palatine tonsil, and lingual tonsil. It may be difficult to locate the pharyngeal tonsil in this figure. Actually, pharyngeal tonsils is in nasal cavity. Pharyngeal tonsils are called adenoid when they become swollen by infection. Swollen means enlargement. Next, Peyer's patches. Peyer's patches are located in the small intestine. They are similar to tonsils. They capture and destroy the bacteria in the small intestine. So, how many Peyer's patches are in small intestine? This figure shows 4 patches. 4 patches? No. This is old paper published in 1965. This paper shows that there are about 60 Peyer patches before birth, and there are about 160 patches in children. They counted patches containing more than 5 follicles. So including patches containing 5 or less follicles, Peyer's patches may be more. Appendix. Appendix is a tube-like offshoot or projection of the first part of the large intestine. Note that it is not the cecum in Japanese mocho. Appendix is chusui, which is projected as cecum. Appendix is also capture and destroy bacteria. Summary. Thymus is T-cell maturation site, and before birth, thymus is also lymphocyte formation site, but after birth, lymphocytes are produced only in red bone marrow. Spleen is blood filter, and spleen destroy worn out red blood cell and recycle iron in hemoglobin, and spleen is a large blood reservoir. Tonsils, Peyer's patches, and appendix are called mucosa-associated lymphoid tissue, MALT. MALT is a sentinel. Sentinel means army in front line. So MALT protects the upper respiratory tract and digestive tract. Okay, do not look at the handout. What is the main function of the lymphatic system? Okay, I will say, first, lymphatic system removes interstitial fluid or lymph. Second, lymphatic system filters debris by physical and biological filtration. Physical filtration means that they use reticular fiber to remove debris. And biological filtration means phagocytosis, eating. Third, white blood cells or lymphatic cells formation and maturation. Fourth, fatty acid absorption in intestine. This may not be the main function, but I somehow include it to make sure. Okay, thank you very much for listening. I'm happy to present to you. Thank you.
Okay, is there any question? Was it very clear? Yes? Are you awake? Thank you very much. It's a very nice presentation. But actually, I like the fact that you also mentioned the names in Japanese. So some of the keywords, if for the Japanese students, if you can mention it in Japanese, I think it's very helpful. Because I think maybe you know some of these terms, but because they're in English and it's very different from Japanese, you may not link them. So that was very good. Thank you.
Thank you.
You spoke very slowly, so that helped a lot. And used very nice graphics and so on. I think you did a very good job. Thank you. So basically, you cover then the anatomy part. So in the rest of the time, we will see mostly about immune system. So just to summarize many of these, you mentioned several of the functions. Lymphatic system, all of them are very good. Yes, removal of interstitial fluid, physical and biological filtration, white blood cell formation and maturation. So if we make a resume, a summary, I would say these are the two main functions. As he mentioned, the last one is not the main function, also from the lymphatic system. So basically it consists of independent parts, the lymphatic vessels, and the lymphoid tissues and the organs. And they are related to the immune system. Okay? The main functions transport fluids back into the blood circulation, the body defense, and obviously with these resistance to diseases. Okay? So we're constantly exposed to different pathogens, right? To bacteria, fungi, viruses, and so on. It's not only when you fall sick that you have been exposed to them. What is amazing about our body is that it's constantly defending us from all these exposures. So the body is constantly in contact with bacteria, fungi, and viruses. And we have these two systems, two defense systems, for foreign materials. Whatever is not from our body is foreign to our body. One is not specific system, so it may react to any foreign substance. We call it non-specific defense or innate system. And the second system is very specific to a specific aggressor or target, for a specific defense or adaptive defense system. Basically, we can organize it as this. We have our first line of defense. The first encounter they have, these foreign substances, materials, or organisms, would be our skin, for example. This is the first defense. The membranes, the mucous membranes that you have, okay? And with these, the secretions, for example, your skin or the mucous membranes will have some secretions. They will protect you. This is the first line. In a second line, if the bacteria, the viruses, or fungi can go through this first line, let's imagine that your skin is not intact, that you have some wounds and cuts, so they will go through. The second line would be some cells, like the phagocytic cells or natural killer cells, some antimicrobial proteins, and especially the inflammatory response. Okay, we will see each one of them in a few minutes. And the third line of defense, if you think like this, very strategic, the third line of defense is the adaptive system. This takes a little more time to react. We have here the lymphocytes, antibodies, macrophage, and other antigen-presenting cells. These two systems, the innate and the adaptive systems, just for educational purposes, we usually separate them, right? But they are working together to protect us, to protect our body. So let's go little by little. First, with the innate system. We mentioned we have the first line of defense, right? We have the skin, the mucous membranes. This is the first encounter that they have, our foreign aggressors. First, we have the physical barrier that we have with the skin and the secretions. The pH of the skin will be in a very tight range. This is on purpose to inhibit the growth of bacteria. Okay. For example, you have some secretions in vagina, in the genital of women, and it's on purpose very acidic also to reduce the growth of bacteria. If you think about your stomach, the mucosa in your stomach also will secrete hydrochloric acid. This will make a very difficult environment for bacteria to grow. And we have also some enzymes, protein digestive enzymes. So you can see we have several secretions with their characteristics. They will protect our body in a first encounter with a foreign substance or a bacteria or whatever we may be exposed to. And we also have not only these physical or environment or secretions that will protect us, but as a second line, we have some cells and some response of our body. We have the natural killer cells, and we have the inflammatory response. He has already mentioned phagocytes. Phago is to eat, so these are cells that will eat foreign materials. Some chemical substances that will kill pathogens, and we have repair tissues, and fever. Fever is also a way of our body to fight for either viruses or bacteria. Okay, so it's a response. Very quickly, let's go a little over each of them. There are some cells that are called natural killer cells, innate cells. They're present in the blood and in the lymph. And this is a group of very aggressive lymphocytes. We have studied very quickly about the lymphocytes as one of the types of white blood cells, remember? So this is a unique and aggressive type of lymphocytes. They can disintegrate, they can lyse and kill cells, kill cancer, particularly cancer cells, or cells infected by viruses. They have other non-specific targets. Remember, this is a non-specific response. They will release some inflammatory chemicals, and they will release these perforins targeting the cell's membrane of the target cells, and will disintegrate the nucleus, resulting in lyse, in the disintegration of the other cell. What is the inflammatory response? This is a non-specific reaction, a non-specific response of our body that is started, that is triggered when our body tissues are injured. And these injuries could be of different types. It could be physical trauma, it could be heat, it could be some chemicals, or it could be an infection. But the main signs, the 4 main signs of the inflammatory response are these 2. The area will become red. If you touch it, the temperature will be rise, so it produces heat. It will be swollen, okay? And of course it will produce pain. So you will be experiencing pain. So these are the 4 main signs that in a particular area is taking place an inflammatory response. Okay? All of this is leading to protection and healing. Okay? So let's see how these signs, how are they produced? First of all, why do we have an inflammatory response? To prevent the spread of these damaging agents or these harmful agents. Okay? Imagine if you have a bacteria or a virus getting into your body, then quickly will be spreading all over. So we need to contain it. We don't know who it is, but we need to contain. So the first reaction will be to contain. So it's non-specific. We need to prevent the spread. It will dispose of the debris and the pathogens, and it will set the stages for repair. So basically, these are some of the main events that will be happening. Okay, the cell will be damaged. They will release some chemicals, especially histamines. These will produce the blood vessels to dilate and to be more permeable. So it will be very leaky. Okay. If they become leaky, as you already mentioned, we will have edema. So more liquid will go into the interstitial space. Okay, this is edema. Some of these proteins are, we saw last week, involved in the clotting process. And some other cells will be also called into action, the neutrophils and monocytes. Okay? So why do we have redness? Because we are increasing the blood flow in that area from the blood vessels that are dilating. So if I have these blood vessel that is dilating, I will have more influx of blood. So it becomes red, and it also increases the temperature, so you will have heat. But if more fluid is going into the interstitial space and you are having edema, this is something not normal. So this will cause also pain and swelling into the area. Okay? So these are the main characteristics of the inflammatory response. Here are the next few slides are in more detail about what is happening. Some of the processes, if this is your blood vessel, your neutrophils are coming here from the bone marrow, and then they start attaching to the walls. So this is called margination. First, leukocytosis, so you will have an increase in your neutrophils. Neutrophils enter the blood from bone marrow, so you have an infection, you have an increase in your neutrophils. They're coming, it's called leukocytosis. They start to cling to the capillary walls, so they start to attach to the capillary walls. This is called margination. And then they become flat and start moving into the interstitial space. This is an important characteristic of them, and it's called diapedesis. Okay? These cells will squeeze out from the capillaries and go into this space. Chemotaxis, because these neutrophils will follow the chemicals that are being released from this site. So it's like calling them, hey, we have an emergency here, so please come to do your job. So they are going here and they are moving into the injured area. Okay. The monocytes, if you remember, they are type of white blood cell, the granulocytes. They will follow the neutrophils, who are another type of white blood cells, and they will go into the area. After half a day, the monocytes will become macrophages. You also have some clotting proteins that are leaking to this area, and they will start to wall off the damage. They will start to contain the damage with fibrin to prevent the spread of the pathogens. The local heat also will increase the metabolic rate of the cells, and this will speed up the defensive actions and the processes, the repair processes. Okay, this is a very oversimplified explanation of the inflammatory response. Another cell that is involved also in this non-specific defense is the phagocytes. Okay, he mentioned already phago is eat. Okay, so whenever you see phagocytes, cytes is cell. So it's a cell that will be eating or disposing of debris or other materials. Phagocytes could be such as the neutrophils or the macrophages. They will engulf the foreign material into a vacuole, into this structure. And remember, they have these lysosomes that have enzymes. These enzymes, once the lysosome comes here and becomes part of this vacuole, the enzymes will be released and will digest the foreign material. Okay, disposing of these microbe, for example, or virus. This is roughly how a phagocyte will do its work. And there's a variety of antimicrobial proteins that will enhance the innate defenses. And these are two of the most important proteins. One is called the complement proteins, and the other one is the interferon. Okay, but the complement proteins is actually not only one type of protein, but it's a group of at least 20 plasma proteins that are circulating in the blood. And they will activate by attaching the foreign cell, either bacteria, fungi, or, for example, a mismatched red blood cell. Why would we be encountering a mismatched red blood cell? Why do you think this will happen? Any idea? A bacteria, maybe we are exposed to the bacteria, we're exposed to a fungi, but a mismatched red blood cell, when can we be exposed to a mismatched red blood cell? From transfusions. But a mismatched transfusion, remember? So we have the ABO blood groups. And so there is this formation of these complexes that are called membrane-attached complexes, or MAC, and they will allow the water to rush into the cell, and it will burst. Okay, basically, that is what will happen with the complement proteins. When the complement, the activated complement is, when the complement is activated, it will amplify the inflammatory response. Okay, but we mentioned that complement itself is non-specific. Okay, that will enhance the effectiveness of both innate and adaptive. And basically, this is the other important part of the antimicrobial proteins, the interferon. There's a long list also of interference, but basically the idea is that these are proteins secreted by cells that are infected by viruses. Okay? They will bind to the receptors in the membranes of healthy cells, and this binding will stimulate the synthesis of proteins that will interfere, and here is why the name, with the ability of these viruses to infect other healthy cells. Okay, so we are stopping the viruses from infecting other cells. And we mentioned also fever as one mechanism of defense. So fever, we tend to think maybe, oh, if I have fever, I have to stop it. But actually fever has a purpose. And the purpose is also to defend the body from infections. But it's a systemic response. What does it mean? It means that our whole body is reacting. Okay, it's not only in one site, but our whole body is reacting, is responding to some microorganisms. Okay. The pyrogens, or these chemicals, are secreted by white blood cells and macrophages. Exposed to foreign cells or substances, these will trigger or start the fever or the process. Why do we need fever? To stop from microorganisms to infect us. Because the high temperatures will inhibit release of iron and zinc that the bacteria needs. Okay, so it's kind of a clever way to stop them. The fever will also increase the speed of tissue repair, and it will increase the metabolism. But so far, this is only our first line of defense. What if these microorganisms can go through our first line? We need more systems in place, right? So we mentioned we have the non-specific and the specific. We have also the adaptive body defense. And there are three very important aspects of the adaptive body defense that make it very unique. One is it's specific for the antigen. Okay, what does it mean? That it recognizes and it will act against a particular pathogen, okay, or a foreign substance. So it's very specific to whom it will react. The other one is that it's systemic. Again, what is systemic is your whole body, okay? It's not restricted to the initial infection site, okay? And importantly also, it has memory. What do we mean by memory? That it will recognize and will have a stronger reaction, a stronger attack from the pathogens that have already encountered before. Okay, so that is what is the memory. So the first time they face the pathogen, they reacted, but on the second, the third, the fourth, from then on, it will have memory of that pathogen. So it will react stronger to that pathogen. Okay, so these are the three very important aspects of the adaptive body defense. It's very specific, it's systematic, and it has memory. Okay, remember these three things. We have these two systems. You may have heard about antibodies, right? Defending our body. We have these two systems, immune response. One is the humoral immune response, which is through antibodies, and the other one is cell-mediated immune response, that is, like the name says, through cells. Okay? We will study them also one by one, but remember that they are linked. There are some overlapping areas between these two. We have the humoral immunity and we have the cellular immunity. Humoral through antibodies. Okay, so the reaction is through the antibodies. And the cellular immunity is through the lymphocytes, so through cells. Okay. So we will react towards something that is foreign, and we many times mention antigens. But what is an antigen? Antigen is basically any substance that is capable of exciting the immune system and provoking a response from the immune system. Okay? Most antigens are large, complex molecules that are normally not present in our body. Some common examples: foreign proteins, acids, pollen, for instance. Okay, when it's pollen season, it's foreign substance. It produces a lot of reaction. But as the immunity system will develop, it inventories, it lists, have a check of all these proteins, so it recognizes them as self or non-self. Okay. But we also have some self-antigens. Okay, these are human cells. These are proteins that are on the surface or some of the human cells. But we mentioned this when we were studying about blood cells types, blood groups. Some we may recognize them as our own, and others may be recognized as foreign. Okay? Our immune cells do not attack, in principle, our own proteins. And I say in principle because we may have some diseases when our body will start attacking our own cells. Okay, but those are out of the healthy or the normal state. Our cells in another person's body can trigger an immune response because they're foreign. Again, think about mismatch transfusions, for example. Your antigens are recognized by your own body as self, but if I put these red blood cells in a person with another type of blood, it will be recognized as foreign. Okay? And this is the whole idea behind also transplants. Okay? Some important cells from the adaptive defense are the lymphocytes. We have read a little bit about lymphocytes. So far we didn't mention what types of lymphocytes we have. Basically we have these two types, the B cells and the T cells. The B cells are the ones that will be producing the antibodies. Okay? And we have also these other cells called antigen-presenting cells. Let's try to go maybe until the end of this part. So we know about the lymphocytes. Maybe I didn't put this. Previously, in the previous class, I put a very, very oversimplified figure, if you remember also, how it goes from the stem cell, the blood stem cell, into the different lines of the blood cells, right? And I mentioned there are also other stages in between, okay? In between the blood stem cell and the mature cells like the erythrocytes or the leukocytes and so on. So the lymphocyte will originate from the blood stem cell, hemocytoblast, in the red bone marrow. And then we will have B lymphocytes. From here, some of them will differentiate into B lymphocytes, others will differentiate or will mature into T lymphocytes. Okay. The B lymphocytes or B cells are the ones that will be producing antibodies. Okay, so they are related to the humoral immunity. And the T lymphocytes or called T cells, they are not producing antibodies like the Bs. Okay, they are involved in the cell-mediated system. Okay, still both of them part of the immune system. Why are they B and T? And this is where you can maybe easily remember. The B cells will mature in the bone marrow, will become immunocompetent, so they will be mature in the bone marrow. Whereas the T cells will be in the thymus. Okay, so this is an important distinction between both of them. Just a few more about the lymphocytes. Lymphocytes are able, they are capable of binding strongly with self-antigens. So I have some cells with some antigens on their surfaces, and my lymphocytes are able also to bind strongly with them. What will happen if my body allows this to continue? Then my body will start reacting towards these cells, right? So they need to be destroyed. Okay, this is called self-tolerance. Okay. Once a lymphocyte is immunocompetent, so it's ready to work, it's mature, it will react to only one distinct antigen, and only one. Okay. How is this possible? Because all the antigens receptors in the surface are the same. So that makes sense, right? If all the receptors are for only one antigen, this can react only to one antigen. And our genes will determine what specific foreign substance our immune system will be able to recognize and to resist. And this is amazing, right? After becoming immunocompetent, lymphocytes in the B or T will migrate to the lymph nodes and the spleen, where they will be encountering the antigens. Okay, so they are ready, waiting there to be encountered with the foreign substances. And the other important cell is the antigen-presenting cells. They actually do not respond specifically to antigens, but they play a role in helping the lymphocytes. What they do is basically they engulf the antigens, and they will present some of the fragments of these antigens into their own surface, where they can be recognized by the T cells. Okay, so what they do is I break down these and I put a fragment in my surface, and this will help the lymphocytes to recognize these. There are different types of antigen-presenting cells. There is no point to memorizing all of them. Just to mention the dendritic cells that are present in connective tissue, in the epidermis, macrophages that are in lymph organs and connective tissue, and some B lymphocytes. Okay. So let's go a little bit more. We have 15 minutes. So we mentioned also we have the humoral immune response. Whenever you hear humoral immune response, you immediately will have to think about the antibodies. Okay, this is antibody mediated. The B lymphocytes with some specific receptors bind to specific antigens. So if I have, this is the red dots. I don't know if you can see it from your handouts. The red dots will be the antigens, and I have here my antigen binding to a receptor in a B lymphocyte. So the binding will activate the lymphocyte. Once the activation takes place, this lymphocyte will start to, in other words, reproduce, to clone. Okay, so I will have lots of them. Undergo clonal selection, and a large number of clones will be produced. This is the primary response. Okay. But there is, most of the B cells clones will become plasma cells, and plasma cells will produce very specific antibodies to destroy the particular antigen that started the production. The antibody levels will peak. This is a primary response, and then it will start to decline. Why will it decline? Because they will not live forever, right? But some of these B cells will become memory cells, okay? And this is how this system can have memory of previous encounters, okay? So at first, I have the antigen. It binds to the receptor of the B lymphocyte. The lymphocytes proliferate. Some of them become plasma cells, produces antibodies specific to these antigens. Others will become memory cells, okay? And in the second response, it could be years later or later, when these cells become again in contact with the same antigen, the reaction will be faster, okay? These clone cells, these memory cells, will produce plasma cells that produce the antibodies and other memory cells. So roughly, this is what will be happening. Okay, so if we can graph this, if we have here the encounter with the antigen, and this is the concentration of the antibody in the blood. At first, we don't have any antibodies towards this antigen, but with time, we start producing antibodies, and we will have the primary response. The antibodies will start to decrease, but we continue to have memory cells. So in a second encounter with the same antigen, we will have a stronger response, which is called a secondary response, okay? We have active and passive humoral immunity, meaning that it could be naturally or artificially acquired, okay? The active immunity, the B cells encounter the antigens and produce the antibodies, okay? This could be during bacterial or viral infections, if they are naturally acquired, or it could be artificially acquired when we receive, for example, vaccines, okay? If you are vaccinated, you are actually inoculated with a pathogen that is either dead or attenuated so that you will not have the disease, but you are exposed to the antigens. And a passive immunity is when antibodies are from the serum of a human or a donor. Okay, so another organism, another person or another organism produce these antibodies. So there is no memory, there is no immunological memory because the B cells are not challenged with the antigen. What you receive is the antibodies, okay, not the antigen. And this is called passive immunity. So the protection will be temporary. This is like borrowing the antibodies, okay? Protection is temporary. It could be naturally acquired, for example, in the case of babies, of newborns. When babies are born, some of the antibodies go through the placenta and will be passed through the baby, okay? And they could be also artificially acquired if we receive treatment like gamma globulin, okay? So this is a figure, acquired immunity for you to remember. It could be natural or it could be artificial. Natural could be active if we have the infection, could be passive if the antibodies passes, for example, from mother to fetus. The acquired immunity could be artificial, active if we receive it as a vaccine, or passive if we receive the antibodies as an immune serum or a gamma globulin as a treatment. Okay. Okay, so maybe this is a lot to take right now. There's only a little more to go, but I think you're maybe a little bit tired. It's about the antibodies, okay? Antibodies are actually proteins, okay? Usually we put them as IG, antibodies, because it's immunoglobulins. They are soluble proteins that we mentioned they're produced by plasma cells, B cells, and they are carried in the blood plasma. So they are circulating in our body. They are capable of binding very specific to an antigen, okay? This is, by the way, a computer-generated image. So when usually we describe the structure of the antigen, we tend to do it like this. We say it has a T or a Y shape because it resembles the letter Y, okay? So it's composed of 4 amino acid chains. These are 4 chains that are linked by disulfide bonds, not very important for you. These are the 4 chains. These are the heavy chains, which are the longer ones, and we also have light chains. Okay. The C is for constant, and the V is for variable. Okay. So each of these 4 chains will have a variable region at one end and a much larger constant region at the other end. So we have a variable region at one end and a constant region on the other end. Variable, constant, constant, variable, variable, constant. Okay, so 4 chains with different sites. And this is very specific antigen binding sites that are present. These are the antigen binding sites, meaning that the antigen will come here and bind to these areas. Okay? So the antibodies responding to different antigens will have different variables, regions. But the constant regions will be the same, or pretty much the same. Okay, so it's these variable regions that will make the antibody the richness that to be able to bind to different antigens. Okay. Why is this? Because we have these types or these classes of antibodies. Maybe you have heard of them. Each class has slightly different roles, but the main classes of antibodies is immunoglobulin M, that works with a complement, immunoglobulin A, that is mostly found in mucus secretions, immunoglobulin D, that is important for activation of B cells, immunoglobulin G, the one that is crossing the placenta, so the newborn will be born with some of the immunoglobulin G from the mother. Immunoglobulin E that has a role in allergies. Not all of them are identical in their 3D structure or their functions. Okay. From this value, you can later on read it with more time. Immunoglobulin B, for example, is most of the antibodies present in your body. Okay, they're responsible for resistance against viruses, bacteria, and toxins. Classically, if we have to describe the structure of the antibody, it would be this figure. Okay, basically it's this figure. Okay, so maybe we can finish. just a few words about the cellular immune response. We already saw about the humoral immune response and now the cellular immune response. Here the antigens must be presented by macrophage. We saw how the antibodies have to bind directly with the antigens. But in the cellular immune response, these antigens must be presented by the macrophage to the T cells. Okay? This is called antigen presentation. The T cells will recognize this antigen as non-self or self. This is a double recognition. And after the antigen binding, similar to what happens with the B cells, it will differentiate in different classes of cells. Okay? We have the cytotoxic T cells, we have the helper T cells, and we have the suppressor T cells. Okay, what they do, each of them has a specialized function. For example, the cytotoxic T cell, we are specialized in killing infected cells through perforins or these inserting these chemicals. The helper T cells will help to recruit other cells, and they will interact directly with the B cells. The suppressor T cell will stop the immune response to go and control, okay, so that it doesn't continue to react. So it will suppress the activity of the T cells and the B cells, okay. So again, this is a summary of the immune response. And only two things about organ transplants and how the immune system plays a major part when we see about transplants. Basically, there are these 4 types of transplants. Whether we receive tissue from another part of our own body, okay, in that case it's called autografts. Do you think in that case the body or the recipient will have any problems with the immune system or the immune response? Any ideas? If you have a transplant maybe of skin from your thigh to whatever area that you are burned, for example, do you think your body will react against it?
No.
No, right? Yes. Isografts, tissues from an identical person. Where can we find an identical person? Only if we have an identical twin, right? If we share genetic material. Allografts, when we take the tissue from another person. It's a person not related, but you have many times heard about matching, right? If another person who's not related to you matches with you, then you can receive donation from it. And also xenografts, if the graft or the organ is taken from a different animal species, okay? Think about pigs maybe or cows. What would be the ideal donors? We mentioned autografts from our own cell and from our identical twin, right? And isografts. So in that case, we will not have any reaction. Xenografts are never successful. Allografts are more successful with a closer tissue match. Just a few notes on this. Just to finish, well, you may have some disorders of immunity, of course. You know very well maybe hypersensitivity or allergies. Okay. Just a few words about these. We may have immediate hypersensitivity, something that happens immediately. For example, if you have an anaphylactic shock, this could be life-threatening as well. Usually it's involved histamine release from mast cells through immunoglobulins. And you also have delayed hypersensitivity. This is when you have sensitivity to an antigen, but the reaction will take maybe 2-3 days to happen. For example, if you have a contact dermatitis, it may not happen immediately. It could be in a couple of days. You may also have some immunodeficiencies when there is some problems in the production or the function of your immune cells or the complement. Think about, for example, AIDS. Okay? Or we mentioned this, your own body may not recognize some of your antigens as your own and may start attacking your own cells. And these will be called autoimmune diseases. There are several autoimmune diseases depending on which of the antigens are not recognized. This is just an example, myasthenia gravis, for example. When your antibodies are destroying some receptors that are in the junction between the nerves and the muscles. So the nerve impulse is not transmitted. Okay? Okay, so this is the end. I think you're very tired maybe from this one. It's rather heavy topic, I think. Many things to review and memorize. Try to review at home once you get there. How was the quiz today? Was it better? I saw you writing a lot.
Yeah, a bit.
Okay, well, thank you very much. We are 5 minutes over the time. If you have any questions, who's the next one presenting? Okay. Are you okay? No problem so far? Okay. You can use this to Riva-san a memo if you want. Okay, just don't read it entirely from your memo, but you can use a memo so that you know what to say next. Okay? Thank you very much. Very good job.
I actually read whole sentences.